Bismillahirrahmanirrahim. Now we start the another paper 4, which is of course a continuation of the previous video, which was the first video on paper 4-2 of the March 22 series, and this is the second video, which will start with question 3, 4, and maybe we can do 5 as well. Starting with question number 3, the red blood cells of people with sickle cell anemia have reduced oxygen carrying capacity. Explain the relationship between the gene mutation that causes sickle cell anemia and the reduced oxygen carrying capacity of red blood cell. Now, this is 5 marks. That's a lot of marks and a lot of stuff that you need to be giving for me to get you so that you get your 5 out of 5. Now, basically, you've got to tell me everything you know about sickle cell anemia. Now, the first thing which will come to your mind is it's homozygous recessive. And what is faulty is the beta globin. What is the cause? The cause is a base substitution. And the base substitution causes a change in the amino acid sequence. When that results, then you can understand what is going to happen. Instead of instead of glutamic acid to valine is non-polar. This causes a change in the 3D structure. Hemoglobin has a lower affinity for oxygen. So hemoglobin becomes less soluble and the red blood cells become sickle cell shaped. Now, as you can see, the marking scheme points are very simple. Sickle cell anemia is homozygous recessive. Then the beta globin is defective. Why? Because there is a base substitution in it. And this causes a change in the amino acid sequence. If you knew what is glutamic acid to valine, fine. But if you didn't know it and you just wrote change in the amino acid sequence, well, you would have got a mark for that. Valine is non-polar, so that causes a change in the 3D shape. Hemoglobin has a lower affinity for oxygen. Hemoglobin less soluble, so they stick together and form fibers. And that is when it is pulled out of shape and it becomes sickle cell shaped. So RBCs become sickle cell shaped, and this was an answer to explain the relationship between the gene mutation that causes sickle cell anemia and the reduced oxygen carrying capacity of red blood cells. Now, the B part of the question is people with sickle cell anemia may need a blood transfusion. One risk associated with a blood transfusion is a condition known as transfusion-associated circulatory overload, TACO, TACO. TACO is caused by a large increase in blood volume over a short period of time, because when you're given a blood transfusion, suddenly the volume is going to increase because you give it in a very short period of time, in a period of 10 to 15 minutes. This increase in blood volume can be harmful. So blood volume 5 liters, suddenly now it is 5.5 liter. Predict the effect of an increase in blood volume on ADH secretion and state one consequence for kidney function of this change in ADH. So now you need to recollect what the ADH do. It's anti-diuretic hormone. When your blood is very concentrated, what is produced is anti-diuretic hormone. Where does it work? It works on the collecting duct. And there is going to be water absorption. So water absorbed, because your blood is concentrated, so the water which was going to go into the urine is now reabsorbed and your blood concentration is normalized. Now it says increase in blood volume on ADH secretion. Now if there is an increase in blood volume, what is going to happen? There's going to be a decrease in ADH secreted. And this is going to reduce the water reabsorption in the collecting duct. So that was the answer which was expected from you for the 1 mark. Or you could have said decrease in ADH and there's going to be an increased volume of dilute urine being produced. So you need to revise ADH if you're not clear about that, and then look at the mark scheme and understand what was expected for you to write. So let's look at the wording. Decrease in ADH, and I said here and reduce water reabsorption in collecting duct. Or you said decrease in ADH and increased volume of dilute urine. So either you said this or you said this, but this would get you that 1 mark. Now coming on to part 2 of the question. One feature of TACO is an increase in fluid entering the alveoli, which makes it difficult to breathe. This increase in fluid can be caused by an increase in blood pressure in the pulmonary capillaries that surround the alveoli. I always say suggest is the most difficult part. Suggest how an increase in blood pressure in the pulmonary capillaries can cause fluid to enter the alveoli. What you have to remember is you have to understand that where would the fluid come from? These are the alveoli, right? What do you have around the alveoli? You have capillaries, blood capillaries. So what is going to happen is blood is going to be forced somewhere into the alveoli. So it's a basic, very common sense thing. Where is the fluid going to come from? It's going to come from the blood into the alveoli. So sometimes it's just a common sense question which you are expected to answer, and you just got to look at it, visualize alveoli, blood capillaries around it. So where is the fluid going to come from? That's just where it's going to come from. Now let's look at the C part of the question. And when you look at such a lot of stuff written, I'm sure you get a little unnerved. I get very unnerved, and I have to read it quite a few times to really comprehend it very properly. People with kidney disease may be at a higher risk of TACO following a blood transfusion. A study carried out in 2019 investigated whether there is a link between kidney disease and TACO, and this study included data from number one, 200 people who developed TACO after a blood transfusion, 105 people who did not develop TACO after a blood transfusion. So we are doing this study on 200 people who developed TACO and 405 people who did not develop TACO after a blood transfusion. So there's a discrepancy. The one who 200 was a smaller number, 405 is a larger number. The people in the study were put into four categories. People with acute means short term, kidney injury, and that was called AKI. People with chronic means long term. He's a chronic late comer to school. For the last 1 year, he's been coming late to school. Chronic means long term kidney disease who did not require dialysis because the body had got used to it. And these were called CKD, chronic, long term kidney disease. So, chronic kidney disease. This was acute, short term kidney injury. So, AKI is acute kidney injury. CKD is chronic kidney disease. But they were not put on dialysis. So please remember that. So their body had sort of adjusted to it and they did not need dialysis. People with severe chronic disease, these were also chronic kidney disease, but these were kidney disease who did require dialysis. So this is CKD plus D. This was chronic kidney disease, but they did not need dialysis. So the body had got used to the high levels of urea in their system and they were okay. But these were the people who did require dialysis. So this is CKD plus D is for dialysis. CKD is chronic kidney disease. And then there was this last group, people who do not have any kidney disease, right? Now in dialysis, a machine is used to carry out the functions of the kidney. Yes, remove the urea. You've studied this in O levels. Dialysis machines, they function and they help in removing the urea when the person, before a person can have a kidney transplant or he is fit for a kidney transplant. Now, what do we have here? We have the results of this study I've shown in figure 3.1. Whenever you have such a thing, I always say take your time and do this question. Either do the other questions first and come back to this question later on because this requires a little bit of thinking. You need to really think, read the question and understand it. The results of the study are shown in figure 3.1. Now, percentage of people is on this side and the categories are here. AKI, acute wali kahani, CKD is the chronic and CKD is the one with the dialysis and this is no kidney disease. Now, what do we have these bars for? This is the one, the clear bars are percentage of the 200 people who developed TACO. So these are the ones who developed TACO in this situation, in this situation, in this situation, and in this situation. And the other color, let me use another color. Now the other is the percentage of people of the 405 people. This is out of the 200 people. This is out of the 405 people who did not develop TACO. So this is in the AKI group, in the CKD group, in the CKD plus D group, and in the no kidney disease group. So you take your time, you study this, but before you study this, you look at the question. The question is, with reference to figure 3.1, describe the effect of having kidney disease on the risk of developing TACO. Suggest reasons for this effect. Now, as I've told you time and time again, you have to develop the skill to do paper 4. You must know how to handle paper 4. This question is never going to come again. But how you handle this question 4 is your strength. And if you don't know that, then I'm afraid all these videos, watching these videos of mine is not going to help you. So please understand, how would we do this question? Now, when you start doing this question, the first thing is you have to describe the effect. And then you have to suggest a reason. So there will be 3 marks for this. and there'll be 1 mark for the reason. So if you give me a whole lot of describe, I can only give you 3 out of 4. You have to give me 1 reason for it as well. So now as I look back at this table, now another thing which you needed to look here, it says describe the effect of having kidney disease on the risk of developing TACO. Now, if you look at this table, we have to only look at the red ones. Why do we have to look at the red ones? Because red ones are the people who develop TACO, percentage of the 200 people who develop TACO. So they're all the red ones. Now look at the red bars. Now, what is the most important thing you'll see here? What is the most important thing you see here is that having kidney disease increases the risk of TACO. Having kidney disease, because these are the people with kidney disease. So there is increased, if you had kidney disease, there is more chance of you developing TACO because all these are more than, but then, okay, what is the next thing? AKI has the highest risk of TACO. You can see here, AKI has the highest risk of TACO. This bar is the tallest bar. This one is smaller and this one is smaller and this is also smaller. So AKI has the highest risk of TACO. Why? Because you see in kidney disease, there is a higher risk of TACO. In no kidney disease, the TACO is there. Percentage of the 400 people who did not develop TACO. So you can see here, they did develop TACO, but they are lesser than that. Then you do a data quote. Whenever they ask you for this, you need to do a data quote. And a data quote is what? It's a very simple data quote. AKI was 32. In AKI, it is 32. This one here is 32. And who did not develop TACO was 18. Who did not develop TACO is 18. This is 18. And this is 32. So if you gave me this, that it is 32 and 18, percentage of people 32 and 18 percentage, and a change in percentage with kidney disease would be plus 14. So this is what you would be expected to be doing this in this sort of a, when you look at a graph like this. I have written all the figures and the 32 and 18, 32 who developed TACO, 18 who did not develop TACO, then in CKD 31 and 21, then in CKD plus D we have 16 and 6. And what is the change in percentage is plus 14. Here I've written in black, plus 10 change in percentage and plus 10 in the CKD plus D. So change in percentage, so you would give me who developed TACO 32, who did not develop TACO 18, and then change in percentage with kidney disease is plus 14. So you would give me this. Then now coming on to back to the reason. What is the reason that you're going to give me? So what is the reason that you're going to give me? The reason is kidney disease reduces ability to remove excess water. More water retained increases blood volume. So reasons is kidney disease reduces the ability to remove excess water. If you have a kidney disease, naturally, it is not going to, one of the function of the kidney is to remove excess water. On a cold day, you drink 5 glasses of water, well, you're going to be rushing to the washroom all the time because this is going to remove that excess water. Because in a hot day, yes, in the summers like these days, it's very hot, so we drink a lot of water, so we will be okay because we're sweating a lot. So the reasons are kidney disease reduces the ability to remove excess water, and more water is retained and increases the blood volume. So this is what you have to give me for the reason. So 3 marks for the effect and 1 mark for the reason. So the first thing that you gathered from the bar diagram was having kidney disease increases the risk of TACO. Then AKI has the highest risk of TACO. So what does this tell you about the skill to do these questions? Number 1, look at the graph. Look at what is a common thing. What is the thing which is sort of coming out very obvious in the graph? Because you must know interpretation of the graph. Then which one is the highest, which one is the lowest, which one is an anomalous reading. So look at the trend, look at the graph and see what is the most glaring thing, what is the most common thing in all this. So having kidney disease increases the risk of TACO. Then AKI has highest risk of TACO. Then data code 32% developed TACO, 18% not developed TACO, and a plus 14% change in the percentage with kidney disease. Now if you look at the reasons, kidney disease reduces ability to remove excess water. They asked for the reasons here. Remember their reasons they asked. So kidney disease reduces the ability to remove excess water, and more water retained, this will increase the blood volume. When water is not removed by the kidneys, of course the blood volume is going to increase, and of course the blood pressure will increase. Now coming to the last part of this question, and this is a 14-mark question, so it's a very intensive question and a very difficult question. Patients who are at a high risk of developing TACO following a blood transfusion can be given a type of drug called a loop diuretic. In normal urine production, 99% of sodium ions in the glomerular filtrate are reabsorbed and 1% are excreted. So in a normal urine, a normal healthy person, 99% of sodium ions are reabsorbed and 1% are gone in the urine. In urine production of people who take loop diuresis, 80% of sodium ions in the glomerular filtrate are reabsorbed. Instead of 99%, only 80% are reabsorbed and 20% is lost in the urine. So sodium ions are lost in the urine. Explain how loop diuretics affect water reabsorption in the kidneys and suggest why this reduces the risk of developing TACO following a blood transfusion. So again, you need to understand, you need to be very clear, and then of course you need to answer this question. So the following points would be the answer to this is more sodium of the filtrate. So less water will be reabsorbed by osmosis. Less water will be reabsorbed by osmosis, so this will increase the volume of urine. And of course, now comes of course because you have to give me how the reducing the development following a blood transfusion. So blood volume from transfusion is reduced. So this was the plus point in it, that the blood volume from transfusion will be reduced. So you had to give me any of these two, and then 1 mark was for this. So the 3 marks are, please read the question very carefully and think what they are asking you and how are you going to answer this. So the skill to do a paper 4 is reading the question carefully and then imagining and putting your knowledge of the entire kidney chapter in this and then seeing how you can answer this question. Of course, the whole thing is what you write in the paper. It is not how much you know, but what you wrote in the paper is actually going to get you a marks or not going to get you any marks. Now coming to question number 4. A part, meiosis is an important process that contributes to genetic variation in a population. Figure 4.1 shows a pair of homologous chromosomes during prophase 1 of meiosis. On figure 4.1, use the letter C with a label line to identify where crossing over occurs. What you have to understand is where crossing over occurs is where non-sister chromatids, non-sister chromatids are going to exchange. There is going to be one half of it, the portion of it is going to break off and get attached to the other one. So it is between non-sister chromatids. So these are sister chromatids, not between these two. These are sister chromatids. So it has to be either with this and this, or with this and this, or with this and this, or with this and this. So you have to see is which is between non-sister chromatids. Okay, now let's go on to the next part of the question. Describe how crossing over produces genetic variation in a population. Now, the easiest answer possible on this earth. Easiest question, how crossing over produces genetic variation. This has come in so many papers and this is just something which you have to just give me all the information which is required, which is crossing over is between non-sister chromatids, then have different combination of alleles, exchange alleles, new combination of alleles, linkage groups broken, generates a unique combination of alleles and random fusion of gametes. If I was in your place, I would have just learned these and I would have just memorized these, because if they come in any paper, you're going to just write all this. There's nothing new that you have to write. So between non-sister chromatids, different combination of alleles, exchange alleles, new combination of alleles, linkage groups broken. If you don't understand any of this, just put it in a Google search and you can figure it out. If you don't understand what is linkage groups broken. You see, linkage is when there are on one chromosome, there are two different genetic information which is very close to each other. And then they will go together. But these linkage groups will be broken because of crossing over. And then gametes have unique combination of alleles, and because meiosis, the function of meiosis is to produce gametes. And then of course there's the random fusion of gametes. Now why is that an important thing? Is because crossing over produces genetic variation. And because there is this unique combination of the gametes, and then there is random fusion of gametes, because after the millions of sperms, which sperm fertilizes that one ovum is just a very random process. And that results in a lot of variation in the population. Part B of the question is again now it's with the genetics question, inherited chain chapter. Wing pattern in the butterfly species Heliconius melopinine is controlled by genes on the autosomal chromosomes, so it's not sex-linked. The gene for banding pattern on the upper wing has two alleles, a dominant allele coding for a full band, so this would be AA or big A small a. A recessive allele for a broken band, so this would be small a small a. A gene for ray pattern on the lower wing has two alleles, a dominant allele coding for rays, so this will be big D or big D small d. A recessive allele coding for no rays will be small d small d. Scientists crossed a butterfly that was homozygous dominant, so big A for both genes. So big D with a butterfly that was homozygous recessive for, so it will be small a and small d small d. With a butterfly that was homozygous recessive for both genes. The scientists wanted to check whether the phenotypic ratio for offspring in the F2 generation. So you see all the offsprings here are going to be big A, big D, small d. This would be the F1 generation. Now, if the phenotype for the offspring in the F2 generation, now when you cross this, when you cross big A, big D, small d into big A, big D, small d, so this is both are heterozygous, then you get a 9:3:1 ratio. This is what we know. We've studied this in the chapter on inherited change. So the scientists wanted to check whether the phenotypic ratio for offspring in the F2 agreed with the expected phenotype ratio of 9:3:1. The results of the genetic cross are shown: full band rays, broken band no rays. So this would be what? You know this. And the F1 generation all would be heterozygous for both. And then in the F2 generation, we have a ratio of 99:21:8:8. Now this is not a 9:3:1 ratio. You can see that. 99:21:8:8. Fine. Now let's look at the next part of the question. Now it says, explain the term F1 generation. So now they're asking you what is the F1 generation. The F1 generation is the generation of offspring from the parental cross. And the parents are homozygous or they're pure breeding. And the F1 generation will be all heterozygous. So this is what you had to give me. The answer to that was very simple, is that you explain the term F1 generation. So the first generation is what? Big A into small a. So all the offsprings are big A, small a. So this is what you had to give me. So the answer to this was first generation of offspring from parental cross. Parents are homozygous. F1 generation will be heterozygous. Then part 2, the scientists used the chi-square test to compare their data to the expected phenotypic ratio of 9:3:1. The formula for the chi-square test is shown. Key to symbols O minus E whole square over E, sum of. Complete table 4.1 and use the chi-square formula in figure 4.3 to calculate the x square value. Now you can see I have done this table for you. It's 25.5 minus 17.5, 306.25 and 12.01. And then you had to calculate the x square value. So 1 mark for you had to do the you had to add all these. So the answer to this, the x square value was 19.45, and you got your 3 marks for that, because you had to get me 3 marks. You had to get, why would we get the 3 marks? 1 mark for the E and the O minus E. So for if you got this correct and this correct, you got 1 mark. And then if you got the O minus E whole square, this correct and this correct, you got another mark. And the third mark is for this. So you must be very clear, why am I going to give you 3 marks for this when I'm checking this paper? Then the question continues and says the critical value at the 0.05 probability level and 3 degrees of freedom is 7.81. Because there are 4 sets of data, it's n minus 1, so it has to be 3 degrees of freedom. Using the result of your calculation in B2, so I've written that here so that I remembered it. Explain whether the results of the study agree with the expected ratio of phenotypes for the F2 generation. So the critical value is 7.81. Your value is more. So if your value is more, what happens? What is the rule? Of course, the null hypothesis is rejected, but of course, we're not talking about that. But it says, explain whether results of the study agree with the expected ratio. So yes, the 19.45 is greater than 7.81. So that's the first thing you write. and then the results are significantly different to the expected ratio. So differences are not due to chance. There is a due to increase in full band phenotypes. So the fact that you wrote that the x square value 19.45 is greater than 7.81, and then the observed ratios is significantly different to the expected ratios. Expected ratios was 9331. This is not 9331. It's very different. So differences are not due to chance, due to an increase in full band phenotypes. You see, the full band phenotypes are much more. If you look at one here, this one, the full band phenotypes are much more. They're 99. And then we come to the last part of this question. Suggest two reasons. Suggest why phenotypic ratios of F do not always match the expected ratio. Well, of course, you know it is linkage. And now that it's in the syllabus, we talk of epistasis, in which they are linked genes. So epistasis was one mark. You could have said linkage. That was also correct. You could have said lethal allele. There could be a lethal allele, which of course results in not overriding all the other effects. Then chance deviations. Then environmental effects. Then you could have said mutations. And of course, you could have also said, because it's a very small sample size, when the small sample size does not really represent. If I say I take a test, I have 100 students in my class and I only take a test of 10 students, well, that's a very small sample size to really talk about the result of the entire class. So it's a very small sample size. And that was all that was required. Okay, that finishes this question. We finish the video here, and we will continue the rest of the questions in the third video. This was the second video, so we'll continue that in the third video. Thank you very much for watching. Please do subscribe. Only question 3 and question 4 have been handled in this video.